Welcome to our biochemistry presentation. Today we are going to look at certain biomolecules, their structure and function, and some key concepts you will find in biochemistry.
Carbohydrates are carbon-based molecules that are rich in hydroxyl groups, carbon, hydrogen and oxygen, hence the name carbohydrate. Carbohydrates are stored in the human body as glycogen to be used as a short-term energy source. Carbohydrates include simple sugars such as glucose and fructose. These are the monomers of the carbohydrate and are referred to as a monosaccharide. The word comes from a Greek translation, monos meaning single, and saccharon meaning sugar, giving us the word single sugar. All monosaccharides are structural isomers, meaning they all share the same chemical formula, 6 carbons, 12 hydrogens and 6 oxygens. Different arrangements make up different sugars. Joining different monosaccharides together also forms different sugars. This is done by glycosidic bonds forming from a condensation reaction where a sugar loses a hydrogen atom and another loses a hydroxyl group. From there, a water molecule is formed and excreted as waste, leaving a covalent bond between single sugars making a disaccharide. Long chains of these bonds form polysaccharides and can make different sugars such as starch. Proteins play varied roles throughout cells or in an organism. Some different uses of proteins include enzymes, hormone signaling, and they can be used for transportation. For example, hemoglobin, carrying substances in the blood. There are 22 different types of amino acids, 8 of which cannot be synthesized by humans and must be taken in through our diets. These are known as essential amino acids. Proteins can also be identified in 4 different types of structure. Amino acids in a specific sequence joined together by peptide bonds to form the primary sequence of different proteins. The primary structure of proteins leads to a unique folding pattern for that specific protein. The interactions from the backbone of the polypeptide chain start local folds and make either the alpha helix or beta-plated sheet. These are the most common types of fold. The overall three-dimensional structure of a polypeptide is called its tertiary structure. Tertiary structure is primarily due to the interactions between the R groups of the amino acid that make up the protein. Proteins are made up of multiple polypeptide chains, also known as subunits. When these subunits come together, they give the protein its quaternary structure. The peptide bonds are formed by a condensation reaction that extracts a water molecule as it joins the amino group of one amino acid to the carboxyl group of the neighbouring amino acid. Lipids are a varied group of heterogeneous organic compounds that are mainly composed of hydrocarbons. They include fats, oils, hormones and waxes. They are polymers of fatty acids and are used for storing energy, signaling molecules, and specific fatty acids are used for the structure of membranes. The most common type of lipid is triglycerides. They come from foods like butter or oils and is stored to be used later by the body for energies. Triglycerides are composed of a glycerol head joined to 3 fatty acid tails by ester bonds. Fatty acid tails can be saturated or unsaturated. Saturated fats are visually straight in appearance and are saturated with hydrogen atoms. They also lack a double bond between the individual carbon atoms. Unsaturated fats have a visual kink in the tail and one or more double bonds in its carbon chain. Triglyceride is formed when 3 fatty acids combine with one glycerol molecule by forming a covalent ester bond in a condensation reaction, releasing 3 water molecules. A hydrolysis reaction is the opposite of this, and the water molecules are added and it breaks up the ester bond between the fatty acids and the glycerol molecule.
Enzymes are proteins that act as a catalyst for biochemical reactions. They speed up the rate of reaction while not changing themselves in the process, meaning they can be used many times. Enzymes decrease the activation energy necessary for biochemical reaction to occur by stabilizing the transition site. This stabilization speeds up reaction rates. Enzymes have specific structures complementary to their structure. They are made up of polypeptide chains of amino acids which determine the structure of the enzyme, including the active site. This amino acid chain produces a complex 3D structure known as ternary structure. Protein structures also include primary, secondary, and quaternary. Enzymes are used to build larger molecules from smaller molecules, change one molecule into another molecule, and break down larger molecules into smaller ones. The active site is the region of an enzyme where substrate molecules bind and undergo a chemical reaction. Every biochemical reaction involves either bond breaking or bond forming. The substrates must absorb enough energy from their surroundings to start the reaction by breaking bonds in the reactant molecules.
Activation energy is a term used to describe the minimum energy needed to activate molecules or atoms so that they can begin colliding with each other, breaking their bonds and producing an exchange of atoms that form new products, otherwise known as a chemical reaction. All chemical reactions, including exothermic reactions, require activation energy for a reaction to take place. During this process, the energy increases to its maximum. This maximum unstable high energy state is known as the transition state, as shown on the top of the image. Because the transition state is unstable, reactant molecules don't stay there long. The energy quickly decreases to that of the products. The arrows in the middle of the image show that the activation energy is the difference between the maximum energy and the energy of the reactants. An enzyme, otherwise known as a catalyst, can be used to increase the rate of reaction and therefore decrease the activation energy. Catalysts do this by binding to a substrate, which allows a reaction to speed up. There are several alternative factors which can affect the rate of reaction by altering the activation energy. These are reactant concentration, surface area, physical state of reactants, temperature, and, as previously mentioned, a catalyst.
Today I will interview two famous scientists on their famous theories for explaining the specificity of enzyme action. First we have Emil Fischer, German chemist, famous for his lock and key theory. I discovered the lock and key hypothesis in 1894, and it can be described sort of like the active site acting as though it's a lock and the substrate acting as the key. The shape of the substrate is complementary to the shape of the active site on the enzyme, meaning when the substrate enters the active site it fits perfectly like a lock and key, and the two bind together forming the enzyme substrate complex. Now hang on there, my name is Daniel Koshland, and in 1958 I adapted the lock and key theory and hypothesised my own induced fit theory. This is where the tertiary structure of the active site changes as the substrate molecule approaches. As the substrate starts to form bonds with the amino acids in the active site, the tertiary structure of the enzyme adjusts so that the active site moulds itself around the substrate. This can be explained like putting your hand in a glove. So the enzyme acts as a glove and the hand acts as a substrate. The enzyme structure is flexible and only complementary to the substrate after the substrate is bound. Thank you. Well, thank you both for your time. In conclusion, to compare the two theories, we know that scientists widely accept the induced fit model. This is because with the lock and key, the shape of the active site does not change when the substrate binds. However, with the induced fit, the tertiary structure of the enzyme changes as the substrate approaches, so the active site moulds around the substrate, meaning it's only complementary after the substrate has bound.
Because of kinetic molecular theory, we know that in chemistry, the more that we heat up a substance, the more particles will move, and therefore the more collisions they will have with each other. This allows for more reactions. There is a similar process in enzymatic reactions, however there is an optimum temperature, and this is at the point where enzymes are working their hardest and producing the most reactions. Beyond this point, denaturation will happen, and you can see this on the graph. We have at the top T, optimum temperature, and beyond this, the gradient of the graph starts to decline. This is denaturation.
Denaturation is the process in which proteins lose their quaternary, tertiary or secondary structures. In the image on the right, we can see a quaternary protein that has been unaffected by temperature, but as our optimum temperature has now been surpassed, denaturation has happened and the protein has unfolded, completely changing its shape. Before, our active site would have looked like this, but after denaturation it will change and the substrate can no longer bind in the same way. This is going to slow reaction rates down.
Our key points are that in higher temperatures we generally see more reaction activity, but beyond a certain point denaturation will happen, slowing rates down, and in cooler temperatures there is less kinetic energy and therefore less reaction activity.
So now we understand temperature, we can look at pH in a similar way. If we think about our graph the same, the x-axis but now as the pH and the y as activity, we will see a graph like this again. This point being your optimum again, only this time it's now your optimum pH level. This is where your enzymes are working at their best.
So unfortunately there is no universal pH optimum. This graph we've plotted is going to look different in every single part of the body. Lipase in the pancreas sits at about 8.0, but lipase in the stomach is about half of that. With pepsin it's 1.5 and maltase 6.1 to 6.8. So even acidic and basic pHs can cause denaturation. If the pH is too low or too high, the chemical structure of the amino acids forming the enzyme are being changed, and we will see the active site denature, and the substrate can no longer bind in the same way. Moving on to inhibitors, they are described as substances that reduce the activity of an enzyme catalyzed reaction. So already we know that we're working with something that is not going to help us speed up our reaction rates. They can act directly or indirectly on the active site, and they can be foreign to the cell or natural components of it.
There's two types of inhibitors, and the first we're going to look at is competitive. Competitive inhibitors can actually fit onto the active site of the enzyme, as you can see here, the blue being the inhibitor. Because this has now happened, they act as a barrier to the substrate, and the substrate can no longer fit into the enzyme as it would before. This will slow down your reaction rate. The second is a non-competitive inhibitor. And instead of competing for the active site, the inhibitor will bind itself to something called the allosteric site, and this is a separate part of the enzyme. Even though this is happening in a separate location, we're still going to see chemical alterations and therefore alterations in shape, and this is going to affect your active site again. So we're now seeing a situation where there's no direct competition with the substrate, but the substrate still can't actually bind with the enzyme as it would have before. Well, that concludes our presentation for today. We hope you've enjoyed it and have learned everything you needed to know. We'd like to extend a warm thank you to guest stars Emile and Daniel. We couldn't have done it without you.